Thank you very much for participating. And once again, welcome to Preparation of Cells and Tissues for Clinical Diagnostics, TEM. Robert Goodwin is the Electron Microscopy Core Manager at the Oxford Instruments Center for Advanced Microscopy, Electron Microscopy Core, the name OCAM EM, at the Medical College of Wisconsin. He holds a bachelor degree in biology from the University of Wisconsin, Stevens Point, and brings over 12 years of experience in clinical research electron microscopy. With extensive expertise in TEM, Robert's work has been recognized in peer-reviewed publications, his most recent contribution appearing in the Journal of Microscopy and Microanalysis in 2024, and he remains committed to advancing microscopy techniques, supporting researchers, and fostering innovation in biomedical imaging and innovation. With that, I'll turn it over to our speaker, Rob. Take it away.
All right, thank you for everyone for attending this. As a talk, I just kind of we're going to touch on what kind of led us up to purchasing this machine and how it has helped in our lab. It's a really great machine. We've processed very small samples, although in small samples, it's very adaptable, so you can process large samples as well. I've been able to do cell pellets, cell cover slips, and all sorts of stuff. We mostly do clinical work here in our lab. We do have a research aspect to this too. So we've started doing we do a lot of renal biopsies in our lab, and it works fantastic for us. Let me move this over here. So as everybody knows, especially if you've worked in a clinical lab, there's usually a quick turnaround time. And especially in our lab, it was up until last year, it was just myself and my boss in this lab. So it's a small lab. And we started getting a huge increase of samples, which I'll show in a few slides here. A lot of the obstacles that we do come into is it's very easy, especially the small specimens, to lose samples. We get samples in at all times of the day. There's protocols that we have to have validated and run by, and then it can take a lot of time, as everybody knows, to process these samples. So it's just kind of an overview of, you can see the various sizes of the tissue. We've got a picture of the robot in there, and it should be stuff everybody's familiar with. This is the paper that Tom and I did up. There will be a link to it in here and then elsewhere in the presentation. So if you wanted to check that out, please do so. There's one more slide for it. So just a general workflow of tissue processing, especially in the clinical sense. We get our samples from the hospitals already in fixative, and just through the entire workflow, and we've kind of abbreviated our standard workflow as well. We've found a few steps that we can shorten and cut out, but it still takes 6 to 8 hours to process a standard sample by hand, or manually, I should say, and then in the oven overnight to embed, and then onto the sectioning and all that. And then same thing with the cell pellets. We spin them down in the centrifuge and continue processing. So you can see here, this is kind of my workload over the last couple years. This is just the clinical side. Fortunately, my numbers aren't showing up on there. But we went from doing about maybe 130 to 150 samples a year in 2018. 2023, it was over 620. Last year, it was right about 600, and this year is going to probably be well up there as well. So we were kind of slow, and then the hospital decided to hire not one but two renal pathologists now, and we are getting stuff from everywhere. And so it presents the problem of how many samples can one person do? And at about the same time we started looking at the tissue processor, my lab director, who was my only backup, was out for several months on medical leave. So I was processing all of these by myself. And we have a pretty quick turnaround time as well. We have a 48-hour turnaround time, and I found the pathologists don't like when you're late. They don't understand that you can only do so many of these in a day. So this processor allows us to keep up with our workflow. Just a sample breakdown of what I mostly do. It's mostly renal, a lot of renal stuff, but we do amyloid, fat, and heart muscle. So we deal with cell pellets, we deal with small tissue samples, ciliary biopsies, muscle, nerve. We can run it all in there. This is a picture of the processor. You can see it doesn't take up much workspace at all. It's about the size of a standard embedding oven, and you have the laptop. It is vented into our fume hood right next to it. So pretty small footprint. Everything's self-contained, which makes it very nice. This is just our layout. So this deck is extremely customizable. Whether you can select which sample or which pieces of equipment that you want to have in there, which ones you want to use, where the chemicals are taken for. We use over where over in the sample section, we mainly use the 96 well PCR plates. But I have set this up to do cover slips. I can do Eppendorf tubes. I can do beam capsules and 5 mil tubes as well, depending on, and it's just a matter of just moving things around a little bit to accommodate it and selecting the proper protocol that you've written for it. You can see in the back where it says Prepmaster 5100, that is the 8-head pipette. There is also a single pipette attached that didn't show up in the picture very well. So you can choose between the two, and it will just automatically select. It'll pick up the pipette tips, and at that point, it's just pre-filling the chemical reservoirs. And we use a combination of aluminum foil. You can get the adhesive cover, adhesive backed aluminum foil, or else in some cases we also use the X-Pierce covers as well. Those just don't like to, they don't they don't hold up to the acetyl and acetone as well. So in the cases where that's used, I just use aluminum foil. So pretty easy to set up. It only takes a couple minutes to get everything set up in the morning, and then from there it's just loading up your samples and hitting go. I wanted to touch base also on the ease of use of the protocol editor. It's really plug and play. This is just a example of what this screen looks like. And in the timeline section over there on the left, you select your steps, and on the main display there, you you just select where you want it to pick up chemicals from and where you want it to deposit. Extremely customizable. Once you navigate through how to do it, it's real easy to create new protocols and change them up. And then this is just another step of the changing up what you want the robot to do when it's doing. So you can see where it says aspirate, it's gonna pick up, my osmium in this case, and it's one column of it. It's gonna move it over to the sample plate and just do one column. Now, if I have several columns, I just select 2 columns or 3 columns, however many columns I need it to do. So it'll have out a preset dispense rates and how much and you can change how much volume per well it's going to pick up. I use 300 mil or 300 microliter pipette tips on there. So I just, and the PCR plates will hold about 200. So you can just change things up. And if you want more or less, it's real easy to do that. You can also have this, program it to, like, for my 70% alcohol. I can have it pick up the water in a certain amount and pick up the alcohol. It'll do a little mix, so I don't even have to have uh, pre-made volumes of percentages of alcohol. This is my protocol that I use. So as you can see, this whole run takes about 2 hours, and that is from receiving we receive ours in fixative already, because We get them from different sources. So that's from the first stage buffer all the way through to my 50/50 resin. We used to do the 100% resin in this as well. It has the capability. One of the pieces of equipment in there will heat up. You can set it to 40 degrees or whatever you like to have your warmer set at. And but we just found it was changing the resin in those little PCR tubes were it's a challenge. So we just run it through 50/50, and then our changes of resin we just do back in the tube that it came in. It's just a little easier that way. But you can see on there, and some people I know are going to be like, you only have it in osmium for 20 minutes. That can't be right. Um, yeah, it's 20 minutes instead of an hour, and you'll see from the photos I have at the end that everything comes out just fine. We did a lot of tinkering with times and really seeing where we can cut things down. So start to finish on one column for me is an hour and 59 minutes. Now, when you add additional columns, it'll add just a couple minutes to each one, just for the time it takes to transfer between the different ones. We also have found too that we can cut out some of our dehydration. I only do 70% and then two changes of 100%. And then we also, same with our resin. I don't do the graduated stages of percentages of resin. I do 50/50 and then into two changes of 100%. So instead of 6 hours, 6 to 8 hours, I can have everything embedded in four, which allows us to do multiple runs per day. And that's just showing that you can do pellets as well in there. Those are cell pellets. The cell pellets we will embed in histogel beforehand, just to kind of keep things intact while everything's moving around. This is just our standard post-processing there, curing, all, all and going sectioning and all that. So the biggest advantages of this is decreased processing times. Uh, we've gone from 6 to 8 hours to less than 4 hours. Uses a lot less chemicals as well. You find, instead of using, 1 to 2 milliliters per sample, you're using microliters. The biggest advantage well, one of the other big advantages was that I can set this up in the morning and it frees me to go and do other things while this is processing. And it's relatively mistake free. If there is a mistake, usually it's because I didn't clip something in the right spot. So it's just, we have a whole checklist that we came up with, pre-flight checklist to make sure everything's good to go. We can do 2 to 3 runs per day in just one shift, and the results are the same. It's customizable. The results are the same, and it's actually been very beneficial. We hired a lab tech with very little lab experience, and you can take somebody with less experience, say, put everything in there, hit go, and they're able to they're able to process without any issues. But the biggest thing was freeing my time up to go run and do other things that need to be done. So I'll get this in the morning, get things going, go sit down, and I'll cut. About the time I'm done trimming, this is done. I need to move it into resin. And then about a little bit after that, they're calling for the second pickup of samples, and then we can get a whole second run going while I'm scoping in the afternoon. So that is going to be especially in small labs where you're on a time crunch, it's very easy to get distracted when you're processing manually. I don't know how many times it's like, oh shoot, I have stuff that I'm processing, and now my 6 to 8 hours is going to take an extra day just because I can't multitask sometimes. So how does it work? There's a big part of this that allows the sample time to be decreased is the shaker agitator set up in there. We have the device that the samples all sit in. You can change it from 300 to 3000 RPM. 3000, it gets a little quick, we found out. But you can also control the temperature on it. There's a chiller unit in there as well. So if you have reagents or anything that needs to be kept cold, you can set that. Or if you need things like your resin to help with infiltration, if you wanted to heat it up, you can heat that up. And there's a little video for everyone to watch really quick. All right, so yeah, that's just a quick overview, quick video. It's basically that pipette just moves the chemicals around. We have used other ones in the past where the tissue is attached to the pipette, so it was very limiting. You could only run 8 samples at one time. This allows up to 96. And then I remember when I first started working here, we had this ancient tissue processor with a color scheme of something that would be at home at the Brady house. It was from the 70s. It didn't even work very well, and I think it used more chemicals than if I were to just process stuff by hand, and it still took the same amount of time. It's an old rotary machine. We got rid of that. Um, let's see, advance. There we go. So this is just going to get into some of the examples that I've done here. All these images coming up have been done on this tissue processor. So the big thing is that I'm showing is that there's no loss of contrast, no loss of tissue integrity. Everything comes out pretty clear, because if it wasn't coming out to how we want it, our pathologists would not be very happy, nor would the researchers that we deal with. So here's just an example of some muscle tissue. You can see all the banding in the muscle tissue. And just kind of a standard renal biopsy. The renal biopsy's contrast is very key. You can see some variation in the in the basement membrane of the capillary loop there. That's one thing that they do look for. But we look for a lot of deposits, and sometimes they can be very subtle. So if I'm not getting the contrast I needed, because things aren't in chemicals too long, or just various reasons, but it's consistently we get the same results. I never have one where it's like, oh yeah, this is not good. Some higher magnifications, these are going to be taken probably around 80 to 100,000 times magnification to see all the structures of the ciliary cells there. uh, so again, very important that we can see everything in that. This is again kidney, since this is the bulk of what I do. This particular one has Fabry's disease, so you can see all the separations in between the membranes in the diseased areas there. And again, very good contrast. Another one that we do real high mag that I need to see the contrast and be able to see clearly is amyloid. And so you can, in here, there's deposits of the amyloid protein, and then at 100,000 times, I have to be able to measure them. So I have to see things clear enough to be able to measure the width of the fibers. And then just some more muscle with mitochondrial disease. Will not, I don't diagnose, I just know that's not right. So I won't get into any details on what it means, but it's just, it looks like a multi-layering effect. And again, just some more muscle. Muscle always looks nice, and so it's good to present that, especially when you can clearly see all the banding and all the structures that you need. A few more ciliary cells, again at 120,000 times magnification, we can clearly see everything that we need to see. And this was this was a panoramic that I did of rat retina. So we have chunks of eyeballs that we were able to press up and cut into small enough pieces, and then tossed it on a slot grid and was able to take a whole bunch of pictures that I was able to stitch together. So you can see all the photoreceptors, rods and cones, and what have you. And then again, this one was pretty neat. As far as a diagnostic aspect, this is a virus. So we're able to see even at 100,000 times magnification, the viral particles. So delicate tissue, big chunks of tissue. It's very customizable and allows us to process pretty much everything that comes through our lab. So just as a summary there, it's the we're able to process small samples along larger samples. There's the other thing that I should mention too is that you can process different samples at the same time. There's no real worry about carryover and contamination, because between each step is so Sam dispensing or aspirating liquids out of the sample holder, it's going to dispense it and in the waste before it moves on to the next one. So it's, if on the rare occasion, if a piece of tissue gets stuck to the pipette, it's just going to be deposited in the last waste bin portion of it. So if it's not the end of the world, we can just always go back, grab that piece of tissue if we really need it, and continue processing manually. But it very rarely happens, and there's no contamination to the other samples. So it's a lot of time saving, allows us to increase our throughput. And in the future, as our samples continue to increase, it will be used. I mean, we use it every day at this point, but it will be probably used on several shifts during the day as we continue to increase. So at our, like I said, we're Medical College of Wisconsin. That's me. That's my old boss, Clive. As some of you in this present, some of you attending may have known him. He is retired now, and we have a new lab director here, Ashish, who is really expanding what we do. So it may come down to we get another tissue processor at some point too. And so thank you. I think that's about the time that we were shooting for. So if we have any questions, I will be here to answer them.
Thank you, Rob, for the presentation on the Prepmaster. I'm gonna look and see if anyone submitted questions. And hopefully all the attendees can submit questions and know where that's at.
Sorry.
So if you have any questions, please feel free, clarification or curious about the ways that Rob went about bringing the device into his lab, or maybe protocol questions, please feel free to submit those. And we have our first question, which is about the waste management. And it's asked, how is the waste managed, especially with regards to osmium?
So everything is deposited into, I can go back into, well, it'll probably be quicker if I go this way. All the waste is separately deposited into its own receptacles. And let me get back to that picture. All right. So on here, you can clearly see where our osmium is deposited. There is a hose connected to this. You can see in the previous picture that this is enclosed. There, and you can see on the side the hose coming off, venting into. There's a big fan that we have in our fume hood. And I shouldn't say big fan, but it's connected to a fan that sucks everything out of there into our fume hood. So there's no worry about fumes. Osmium is deposited in the center row here, in the covers. So we have it in, this is where it's going to pick up. I'm assuming you guys can see my pointer. So it's going to take it from the chemical reservoir, move it to our sample plate, and then deposit it in the waste receptacle back here. The osmium goes into its own collections row there. I have another one in there for buffer, and so everything's separated. And then I have a bulk reservoir in the back here for just the water, acetyl nitrile, and ethanol that I use. And so all we do is, it's when we're done, we pipette it back into our waste bottle, which is then collected by our environmental waste management people here. So hopefully that answers your question.
Thanks, Rob. The next question we have is, if you need to prepare a sample with a certain direction or orientation in the resin, do you do the orientation of the sample once you pull it out of the machine?
Yeah. So these are just little conical tubes. So before I start the process, I make sure that everything's sitting down in the bottom of the reservoir. Sometimes you'll get a little air bubble that you got to tap out of there. But then, yes, for instance, the renal biopsies, I have to be in a certain orientation. Muscle has to be in a certain orientation. So those ones, I just prepare that once I embed them, that's when I orientate them. If I have something that's not elongated, I should say, like a cylindrical shape like my renal biopsies are, if I still need it in a certain orientation, I'm going to cut and prepare that sample so that after, it's just as if you were processing in a 5 mil tube or something like that. I'm going to cut it in a way that I can tell orientation. We've done rat corneas, just the cornea, the outer portion of the eye, which that was a little bit challenging. It processed well in here, and then I just cut those into a shape that I can tell, yes, this is, I can, it just makes it easier for me to identify which way it needs to be orientated.
Thank you. Okay, our next question is, could you elaborate more on how cultured cell pellets are processed?
Sure. Yeah, we don't, so we receive stuff from other labs that are already grown on the cell pellets, and we have, it's not in this picture, but I have a cell. I think it's got, I forget how many holes it has in it, but it's just another plate like this that has round wells in it that the cover slips in. And so for those, I don't agitate. Obviously, I don't want to shake them off or anything. So I just place it so in my protocol, I have it instead of my sample area being on the dock agitation station, my samples are just in the number 2 slot. So I pop the plate in there, they're on the cover slips, and then I have it dispense more liquids because they tend to be a little bit hydrophobic when you're dispensing, and I have to have more liquid in it. So as I go through and edit the protocol, I make for those adjustments. And then since it's not agitating, I do have the times increased a little bit for those. And then the only thing is that if you're going to do those, a lot of people tend to bring them in polystyrene plates. If you're going to do anything with acetone or acetyl-nitrile, you need to have the polypropylene stuff because they don't like to play well together with the polystyrene anyway. So polypropylene and all these supplies, everything you see in here, you can get right through EMS. When you click on the link for the Prepmaster, there's a drop-down menu at the bottom, and they have all the supplies ready there for you to order from them.
Thank you. And the next question is, do you use vegetable oil to deactivate your used osmium?
Yes. Yes. So in my fume hood, there's a waste jar with vegetable oil, and it just goes in there. There have been we were in one of the talks that I listened to, people are using aspartic acid, I think it was. I did talk to but that is just completely up to whoever collects your chemicals. I talked to the people here about using that, and they're like, we just prefer you to use the vegetable oil. So yes, it goes in vegetable oil, and then my chemical people come and pick it up. And so even in this case, you can take and if you're worried about any contamination or anything, you can fill those reservoirs or put a larger reservoir. So in these ones covered in tin foil, those are 30 milliliter reservoirs in there. There's 4 of them. I just cover them to keep evaporation down. But you can have it deposited in the osmium and vegetable oil if you want. And so you can take care of that step right away.
All right, we have a question from the audience. I'll allow them to talk. Hasib?
You're muted, Hasib.
Hello. Is there
Hello?
any vacuum desiccator in this machine?
I'm sorry?
Vacuum desiccators. If you
Vacuum.
need a vacuum. Yeah.
Oh, you guys are what are we?
He's asking if there's vacuum desiccation capability. So can you put a sample under vacuum on the machine?
I don't know. That would probably be a question for Tom if he wants to chime in. It's nothing that I do in the lab here.
No, we don't have anything off the shelf to do vacuum on the machine. We could perhaps build something custom.
Okay.
And so that is one of the great things about this machine as well. Like I've touched on the customizability. Tom, I'm fortunate that he lives about an hour from where I work, and so we communicate a lot. And there's a lot of back and forth of saying, "Hey, we might need to do this, or this isn't quite working. Maybe we should do this, or here's an idea." And and I've sent him most of the protocols that I've typed up or done up. So, and I'm open to sharing. So anybody that does get started with this has some pre-made ones, and it's just a matter of tailoring them to your needs. But he's very capable of seeing what people want the capabilities of this to do and what can we do with it.
Which angle we can rotate the sample while processing?
To rotate it?
Yeah.
No, I mean, you can pause the.
There is agitator or rotator also. Is there availability?
No, there's no rotation of it, but you can, if you need to. I mean, it's agitated, and while it's not moving a whole lot, it's the chemicals are moving in there. So in a sense, it is rotating. But it's very easy. If you need to do something while it's running, you just hit pause, and it's going to pause wherever it's at in there. So if you need to pull the samples out and move them. But that agitator, I have a large rotating machine that makes a lot of noise in my fume hood. That the agitation takes the place of the rotation. So if you need your samples moving while you're processing them, that's what the agitation is doing.
Okay, so thank you.
Okay, thanks. So moving forward, we had a question about heating certain chemicals. Is this part, if it's part of a protocol, can you set up your chemical in the reservoir and do some heating? Or
Yeah,
cooling?
yeah. So it's, for instance, I mean, when we were doing the resin in here, we'll put them on at 40 degrees. I don't recall how hot the agitation station goes. This would probably be questions for Tom. But then also where the dehydrants is, I know we can set that to a certain temperature as well. So there is temperature control in these.
It goes up to 100. We rarely go above 70.
Okay, great. There was a comment about the osmium neutralization. Ria was saying that they use corn oil. Used to use corn oil, but now they use milk powder and water, which neutralizes osmium, and so they just made a comment.
Yeah, I've heard that too, and I've brought it to the attention of our chemical waste people, but they're like, so they just want us to continue using the corn oil. So they're the ones that pick it up and dispose it. So I'll do what they want to do.
And there is one more option that some people are doing is just putting their ethanol in the osmium, and that does the deactivation for you as well.
Okay, and we have a question from the audience. Anurag, go ahead.
Hi, Robert. Hi, James.
Hi.
It was very good presentation. And Robert, I have a question. Do you have any images of manual processing, as you have shown the images processed samples by your tissue processor? So just for comparison.
Yeah, I mean, I could probably. I think I'm still sharing here. If I go back a few years, let me just see here. Get to see the inner workings of my messy computer. There's nothing that you can't see in here. So say if I go back to 2019, I can probably just pick any one of these. Now we'll do the next year because I know when we were really hitting hard on the renals.
I don't know about anyone else, but I still see the slides.
Okay. Oh, because I'm not here. Let me 1 second. How about?
And after this, I have one more question.
Let's see.
Yeah, okay, that's better.
Yeah, let me move this over. So yeah, I mean, this is, and it's one of these things. It can very well come out a lot different. I mean, this wasn't the most pristine sample to begin with.
I can see.
But it's you can still see comparable contrast. These were taken on our old Hitachi. So it's about shortly after we got a new microscope, we picked up the processor as well. So a little bit newer microscope. This was on a microscope that was about as old as I am, and but we haven't seen any issues with contrast, any degradation of the tissue, because that was my biggest thing. I mean, and I was very skeptical at first when, Tom says, "Oh yeah, you can process these things really quick." It's like, but
Mhm.
we've only got it in osmium for 20 minutes. I'm not going to have the contrast, or it's moving through the dehydration
Yeah.
phase really quickly. How is that going to, it's not going to embed very well. But that agitation really does. I mean, it's taking, like I said, taking place of the rotation. And
Yeah.
we're finding that in certain
Yeah. There
areas,
is a. Yes.
yeah, we're just finding certain areas that, we don't have to do the 50%, the 70%, the 90%, the 95%, the 100%, the 100% in my dehydration phase. I've tested it, and
Okay.
about the only time that I found that you really do need to elongate or prolong the dehydration phase is with nerve biopsies. They tend to be a little more resistant, and the ones that we get are very large as well. So they're very rare. We don't get them a whole lot, so we'll still process those ones by hand, but we'll elongate the dehydration phase on those. But yeah, 20 minutes in osmium, and you can see the contrast that we were getting is just fine.
Okay. Okay. I have one more question. Can you hear me?
Yeah.
Mhm.
Do you treat a uranyl acetate before discarding or before handing over to the outsourced people? Do you treat osmium tetroxide to make it neutralized with any chemical?
So the urinal acetate, no, I just have a waste bottle. Osmium tetroxide, I put in corn oil. And we have a whole department here that handles with the chemical waste. So they handle with
Okay.
disposing all of it. So what they do with it, I do not know. But I know
Okay. Oh, yeah.
depending on what country you're in, the urinal acetate, there's a lot of regulations on whether or not you can get that or not. I know in some places, I think they use lanthanum. We have tried that as well just to see, but we still had better results
Mhm.
with the urinal acetate.
Okay. I think that is called uranyl free or uranyl less,
Yeah,
if I'm not wrong. Yeah.
yeah, yeah.
Okay.
So I know some countries can't get the urinal acetate. Um, we can
Yes,
still get it. We can get it
yes,
here, and
we
I got a
use uranyl
lifetime
acetate.
supply. So yeah, no, I don't know what I just have a waste bottle that I put mine in. I don't pre-treat it with anything. They come and pick it up, and what they do with it, I don't know.
Okay. Oh,
All right, and I think if there are no more questions, then I will tell you one last thing, which is that, if I get to the back of the slides here, there is a survey that will be sent out at the end of the webinar.
Too far.
And
Actually, James, I got one quickly if we can cover this.
oh yeah,
Something's always intriguing about your dehydration, Rob, which is acetonitrile. And everyone has done ethanol, and then people use acetone sometimes, and then transition solvents. How did you end up using acetonitrile?
So we use methanol instead of ethanol here, and then from from what I was told when I started working here, it's just a little more stable. But then as far as the acetyl nitrile, we use it because it has a lower evaporation rate than acetone. Um, and so you can see the images, it doesn't affect anything. And especially in the tissue processor where I don't want things it's because these chemicals are sitting in those reservoirs for the entire time. And so I don't want things evaporating while they're sitting in there. Acetonitrile has a lower evaporation rate. And from what I was told, was a little bit cleaner than acetone to use.
Thanks.
oh, and maybe we have one more question that came in. And the question is, have you ever needed to combine osmium with potassium ferricyanide to help with contrast? Or does osmium only, is that good enough?
We still do on occasion, just depending on the tissue. All our muscle tissue we used to. I just kind of stopped doing it. It didn't really seem to make a huge difference in what I was seeing. There, on the research side, there are some protocols where we do still use it. I'm not gonna. I don't handle much of the research stuff, but it is something that comes up now and then. I think it was like 0.03 grams in 1 mil, so it was a very small amount. But yes, and then also you can, in certain cases too, you can add tannic acid to your buffer stage to increase some contrast as well.
Thanks, Rob.
But yeah, we used to, on all our muscle samples, we used to do that, and then we kind of just started looking, and it's like I'm not really seeing a huge difference to justify adding that step to it. So because it was kind of tedious weighing out pre-measured amounts of very small amounts of all that. But we do still use it on some occasions.
And I will add that if you are doing complicated protocols, perhaps you're not in a clinical setting, but you might run a core facility, or you have maybe you're doing volume electron microscopy, which tend to be very long protocols. The system, like the Prepmaster robot, can definitely handle a lot of different steps. Maybe you have osmication, ferrocyanide separate, you go back in osmium, tannic acid, thiocarbohydrazide, UA, lead aspartate, that whole long series of steps that tend to come alongside volume EM. You can run that on the Prepmaster, no problem. So it's intended to save you time with those kinds of workflows. But Rob, with that, I thank you very much for your time and for your presentation today, and Tom for your participation in the panel, and to all of our attendees for joining and asking questions and hanging out for that period. If you complete our post-webinar survey, which will come to the email you registered with, you can get $100 off your order of anything that exceeds $500 or more at our website, emsdiasum.com. And if you are interested in the Prepmaster and would like to learn more, either a virtual demo or to chat with someone like myself or Tom, please feel free to, we'll follow up with you at the end of this webinar, and feel free to let us know. And anything else you'd like to add, Rob or Tom?
Just on that also thank you guys, EMS, Tom, everyone making this happen, putting it together. It was a lot of work. And then, yeah, if anybody needs to if any other questions come out, you can reach out to me through email. And likewise, I've also told Tom and the folks at EMS, if you're in the Milwaukee, Wisconsin area and want to kind of see it in action, just shoot me an email, come up, visit my lab, let you look at it, answer any questions.
Thank you. Yeah, that's a great offer. So if you want to go check it out in person, Rob will host you. So,
and I just want to say thank you very much, Rob.
Yeah.
And with that, I hope everyone has a great rest of your day or evening, wherever you might be, and take care. Thanks.